All right, well, welcome to lecture number 4 of Biochemistry and Society, which is complex biomolecules. So in this lecture, we're going to talk in more detail about actual proteins, nucleic acids, lipids, and carbohydrates. So we finally are going to devote an entire lecture to these 4 main classes of biomolecules. So we'll start out with proteins, which are polymers of amino acids. And then we'll talk about what different protein functions are, how they fold into their 3-dimensional shape, and everything like that. So proteins are very important. So we'll spend most of the lecture talking about proteins. Then we'll talk about the nucleic acid polymers, DNA and RNA. So we'll talk about the structure of these different biomolecules, how they function, what they're doing in your cell, all sorts of interesting stuff. Then we'll talk about complex carbohydrates. So these are polysaccharides, so long chains of glucose or other carbohydrates that are really important for structural support in plants, structural support in fungi and in insects, and also in energy storage in the form of glycogen and in animals, and then starch in plants. And we'll talk a little bit about protein glycosylation as well. And that's where you can add carbohydrate groups onto different proteins. And then we'll end up talking about lipids. So lipids actually don't form polymers, but they form cell membranes, which can sort of be thought of as an aggregation or a polymer of lipids. But cell membranes are really important as well. So we'll talk about how the chemical structure of the lipids forms these cell membranes and why they're so important to biology and biochemistry. So again, it's a lot of information in this lecture, but you don't need to remember anything for a test. So just sit back and listen, and hopefully you find it interesting and learn some stuff. So let's get started. Lecture number 4, complex biomolecules. So as we discussed before, there's 4 main classes of complex biological macromolecules. So they are proteins, which are polymers of amino acids, which we discussed in the previous lecture. Nucleic acids, so DNA and RNA are large nucleotide polymers. Carbohydrates, which can be formed from large polymers of very simple sugars, specifically glucose and others. And lipids, which can actually, don't form polymers, but different lipid molecules can assemble to form membranes, which you can sort of think like a biological macromolecule. So these are the 4 main classes of biomolecules, and we'll spend this whole lecture talking about each one individually. So we'll start out talking about proteins, because proteins are arguably the most important biomolecule in biochemistry, or they do so many different things in your body and in biological organisms in general. So proteins are amino acid polymers. So you have these individual amino acids here, which we talked about last time. There's an amino group, a carboxylic acid group, then there's an alpha carbon, a hydrogen, and then a specific R group. And so what can happen between these 2 individual amino acids is that this carbon group can actually form a covalent bond with the nitrogen group on a different amino acid. And then you lose a water molecule, so an H2O will come off, and you can form what's called a peptide bond. So that's shown right here. So this is a covalent bond between one amino acid, and it connects the carboxylic acid group to the amino group of another amino acid right here. So this peptide bond can form what's called polypeptide chains. So this is going to be a bunch of different amino acids linked together by these peptide bonds, as you can see right here. So on each of them, the carboxyl group is attached to the nitrogen group, carboxyl group, nitrogen, amino group, carboxyl group, amino group, carboxyl group, amino group. And so in this way, you can link a bunch of different amino acids together with the emphasis on the word different. So you can have a lot of different amino acids. So here's a tyrosine right here, a glycine, which is very different chemistry, another glycine, a phenylalanine, which is similar to tyrosine but a little bit different, and then a leucine, which is another hydrophobic amino acid. So you can get all these, basically an unlimited combination of the different types of amino acids that are linked together in these polymers to form proteins. And this, we'll talk about this more in a minute, but the specific sequence of the amino acids in that polypeptide polymer that forms the protein is actually going to dictate both the structure, so how the protein folds, and its function. Most proteins, so these polymers, these amino acid polymers that make up proteins can be extremely large. So the largest protein known is titin, which is like tens of thousands of amino acids in a chain. But that's sort of, that's a very unique protein. Most proteins are about 400 amino acids long, some a bit larger, some a bit smaller, some way larger, and then some way smaller. So there are a group of things called oligopeptides, or just peptides. And these are just peptides made up of not that many amino acid chains in a polymer. So angiotensin is one. So that's just made out of 8 protein or amino acids linked together. So that's really important in the kidney to regulate blood pressure. Vasopressin is another one, also made out of 8, and that's secreted in the pituitary gland to mess with blood pressure, and it's an antidiuretic, so you actually retain water, so it sort of controls the urinary system. Oxytocin is another one you might have heard. So that's just a small signaling molecule. All these are signaling molecules so far. And it's only 9 amino acids long. And that one is involved in milk secretion, uterine contraction, and also maternal behavior. So it's actually called the love hormone is what it's been called. And there's also an example of melatonin, which is 26 amino acids long, which is still very long. And this is actually, if you get stung by a honeybee, this is the peptide that's going to sort of mess with, it breaks cell membranes, so it can cause, lysis of cells. And then there's defensins, which are, 28 to 42 peptides long. And those are just basically antimicrobial peptides. So when they're in proximity to a bacteria, they can cause that bacteria to lyse. Anyway, so this is just some example of some very small peptides, 8 to 40 amino acids long. But most proteins are basically polymers of these amino acids that are around 400 long. They come in all shapes and sizes, but that would sort of be the average amongst most proteins. So what's very interesting about proteins is that the specific amino acid sequence in that protein actually contains all the information of how that protein is going to fold in 3 dimensions, and then based on that, what its function is. So that's a pretty interesting sort of thing that was sort of a surprise to scientists when they first figured that out. You could basically take a protein, completely unfold it by just adding chemicals to it, take those chemicals out, and the protein would refold in exactly the same way, just based on the sequence of the amino acids in that polypeptide chain. So the sequence of the amino acids is what's called primary structure of a protein, or each different protein is going to have a different order and types of amino acids. So this specific one has proline, lysine, valine, lysine, alanine, histidine, glycine, lysine, lysine, valine, leucine, glycine, alanine, phenylalanine, serine, aspartate, glycine, leucine, alanine, and histidine. And maybe it'll go on longer than that. And these are all linked by these peptide bonds between these 2 amino acids. So these are covalent bonds. The individual amino acids in this in this primary sequence. So this will first be expressed as just a big long polypeptide chain. So now it has to fold. So the first part of it folding is that the individual amino acids will interact with amino acids nearby them, or generally nearby them. And that will result in what's called secondary structure. And so these are really stable forms for that polypeptide chain to fold in 3 dimensions. So there's 2 kinds. One's called alpha helices, and another one's called beta sheets. And so these are super common features of basically almost all proteins. There are some exceptions. But the example shown here is an alpha helix. So the peptides will interact by hydrogen bonding interactions in the backbone chain and form this helix like you can see right here. So these amino acids are turning and they're interacting with each other via the backbones. So the reason why this is so stable is because these side chains, a lot of times which are really bulky, are able to stick out from the outside of that helix that just formed. So that's very important. So that's a really stable thing to have these bulky side chains sticking out away from this central helix. It makes it really stable. So now that these side chains are sticking out from these helix, they can interact with other parts of the protein. Let's say there's another alpha helix here. They can interact with side chains sticking out from that secondary structure. And then what will happen is those interactions between the side chain and other things as well will cause the overall fold in 3 dimensions of the protein. So of that polypeptide chain. So here you can see there's a bunch of amino acids in this protein. You can see alpha helices. There's a bunch of alpha helices in this protein, and the side chains are all kind of interacting with each other. And hydrophobic ones are going to be in the inside. Hydrophilic ones are going to be on the outside. And overall, you get the full, that's how you get the full folding of the protein. And again, that folding is what's going to dictate what kind of job the protein is going to do in your body. Now, there's one last part of protein structure, which I have to mention, which is quaternary structure. So there's a lot of instances of different proteins where actually multiple polypeptide chains have to come together to get the fully functional protein. So this protein right here is called hemoglobin. It's what actually transports oxygen to your blood cells. It's what makes your blood red. So it's not functional unless there's these 4 separate folded polypeptide chains that interact. So only when all 4 of those are present is this protein going to function as it's supposed to. So those are the different levels of protein structure that give rise to the fully folded protein. So we have these amino acids that are linked together covalently in a specific sequence. Those amino acids will interact with adjacent amino acids to make this really stable secondary structure. The side chains will interact with their environment and with each other to make the folded structure. And then sometimes, quite often in proteins, they'll need multiple polypeptide chains to come together to have the full function. So, but take home is that these polypeptide polymers of amino acids will fold into a 3-dimensional protein. So as I said before, amino acids that are kind of close to each other in that primary sequence, so in the amino acid polypeptide chain, they will actually interact by their backbone amino and carboxyl groups to form secondary structures, and one of those is called an alpha helix. So alpha helices are formed, so what you have is a polypeptide chain here. So they're going to fold over where this carboxyl group right here, which has this oxygen. So that oxygen is going to have a partial negative charge. That, when it folds into this conformation here, into this helix, that's going to make a really strong hydrogen bond with this partial positive charge on this amino hydrogen right here. So that'll happen between all sorts of different residues that are generally 3 or 4 amino acids away from each other. So this interaction of these backbone hydrogens results in this helical structure. And so what's that's caused by is you can see here's the amino hydrogen group right here, and it's going to hydrogen bond with this oxygen group here. Here's another oxygen group on an amino acid. It's going to hydrogen bond with a hydrogen on the nitrogen of this other amino acid. Same with right here and right here. So those backbone interactions, those backbone hydrogen bondings make this helical group here that I said is really stable because the side chains, these sometimes bulky side chains like tyrosine and tryptophan and isoleucine and all that stuff, they're going to stick out away from that alpha helix. So that's a really stable structure. So a lot of times, most of the time when we look at proteins, they're going to look something like this. So this would be a protein right here that is made basically entirely of these alpha helices. It's sometimes also alpha helices are depicted as a column or like a cylinder. And so that would be like right here. So here's actually a membrane receptor, and it has alpha helix, 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 in and out of that membrane because there's hydrophobic side chains sticking off from these alpha helices. So the alpha helix is commonly seen in proteins. I mean, almost most proteins a lot of times have alpha helices or the other form of secondary structure, beta sheets. And it was actually discovered, it was figured out by this guy, Linus Pauling, who's sort of a huge figure in biochemistry. He was a really important chemist. He figured out a lot of information on orbitals, on how electrons move around atoms. He also figured out this alpha helix structure in proteins, and he actually made a mistake and thought the DNA double helix was a triple helix, and that allowed Watson and Crick and the others to sort of swoop in. But he's the only person to ever receive actually 2 Nobel Prizes as a solo Nobel Prize. So that's Linus Pauling right here. And so he discovered these alpha helices, which are really important to protein structure. When I show you proteins from here on out, don't be surprised if a lot of them have this ribbon structure here. That means there's an alpha helix at that position. So the other secondary structure that's really common in proteins is the secondary structure called beta sheets. And so those are similar to alpha helices in that it's these backbone hydrogen interactions. And it is by sort of adjacent amino acids on the chain, but they can be kind of farther apart. So what happens in these beta sheets is sort of what's going on right here. So you have this polypeptide chain right here. So here's the alpha carbon, here's the carboxyl group, this is the peptide bond right here, here's another alpha carbon, here's a carboxyl group, peptide bond right here, alpha carbon, and so I'll just circle the alpha carbons. So this conformation in this protein is really stable because just like what we saw with the alpha helices, those bulky side chains are sticking out from those alpha helices. In the beta sheets, same sort of thing is happening where one of these side chains is pointed below, the next one's pointed up, the next one's pointed below, the next one's pointed up, and the next one's pointed below. So that would be called a beta strand. Now, the backbones of these beta strands can actually interact with each other, and then that makes what's called a beta sheet. So it can happen in 2 different ways. There's beta sheet right here, beta sheet right here. So these are both 2 beta strands. So 1, 2 beta strands, 1, 2 beta strands. And they can actually interact and hydrogen bond with each other to form what are called these beta sheets. And so these are secondary structures that will also be found often in proteins. So they're depicted as sort of these arrows with directionality. And that just means that the amino acid chain is going in that direction right there. But a really cool thing and why these are so stable has to do again with those side chains. So right here, the side chain is pointing away. And this one, it's pointing above. And what's the next one? This one, it's pointed away. This one is pointed above. This one away. And same with this one. Above, away, towards, away, towards, away, towards, away. So it's a really stable structure, and it can be found in a lot of different proteins. It would be depicted like this, and you just have to understand that the side chains are sort of coming up and below from this beta sheet right here. And so that makes a really stable structure. So beta sheets are the other, apart from alpha helices, the other really important secondary structure found in proteins. So these alpha helices in these beta sheets on the protein, these side chains are going to be sticking out from either side of those. So those side chains will then interact with other molecules and other side chains or backbone molecules to actually form the fully folded protein. So right here we have a molecule myoglobin. So you can see it's made out of basically alpha helices for the most part. But then these alpha helices sort of fold into a specific structure. So you can depict that in this way, where you just show the alpha helices. There's also a heme molecule here, which actually carries oxygen for this protein. Or you can depict it as sort of this surface molecule, where you just show the space that the individual atoms are filling up on the protein. So it gives it a more solid sort of depiction. But you'll see proteins depicted in both ways. Now, one really important thing, so the side chains are going to interact between these different on these different alpha helices in this specific example. The other really important thing is that for the most part, a general trend is that hydrophobic amino acids, so hydrophobic side chains, are going to be found on the inside of the protein. And then hydrophilic residues are going to be found on the outside of the protein. And that just makes perfect sense because hydrophilic residues, like the polar ones, the positively charged ones, the negatively charged ones, those can interact with water in the environment. However, the hydrophobic amino acids like leucine, isoleucine, phenylalanine, tryptophan, those are going to be buried within the protein because they're not going to want to interact with water. They'd rather interact with each other. So in water-soluble proteins like this one, the hydrophobic residues are going to be clustered at the inside of the protein. Now, this is actually flipped for proteins in the membrane. We'll talk a bit later in the lecture about proteins that are in the membrane. But when a protein's in a membrane, so this right here is a membrane protein. And so it'll actually be embedded within that hydrophobic membrane. And so it's actually going to be within that membrane, and then there's going to be a pore that water can actually flow through this protein. So in this instance, those hydrophobic proteins are actually going to be embedded in the membrane, and then these hydrophilic residues are going to be in the inside and the outside of the protein. And actually in that pore region where water can go through, that's going to be more likelihood of hydrophilic residues. So we talked last time about the amino acids, how they have a lot of different flavors. Some are hydrophobic, some are hydrophilic, some are charged. This really comes into play when you're thinking about overall protein folding. So those hydrophobic residues in general in water-soluble proteins are going to be in the inside, and those hydrophilic amino acids are going to be on the outside of the final folded protein. And that actually helps dictate the fold of the overall protein. Now, another really common feature in proteins that helps mess with its fold are these things called disulfide bonds between cysteine residues. So cysteine amino acids are a little bit special in proteins. So they have these sulfur groups. So it's a sulfur and a hydrogen. So this is one cysteine. This is the other cysteine. You see it has this sulfur and hydrogen group. So under the right conditions, so under oxidizing conditions, which you don't need to worry too much about, these two sulfur atoms can actually lose those hydrogens and the electrons associated with them and form a covalent bond between these two sulfur groups. So these cysteines might be really far apart on the protein, but they can still, if they're in close proximity to each other under the right conditions, they can still form covalent bonds either within the same protein or in a different protein. So this is a small protein shown right here. And this cysteine residue right here and this cysteine residue are actually forming a disulfide bond. Same with this cysteine and this cysteine, they're forming a disulfide bond right here. And then this cysteine and this cysteine are forming a disulfide bond. So these cysteines are actually located far apart on the actual sequence of the protein, but when it gets folded, they come into proximity and then you form those really strong covalent bonds. Now, a really important and medically applicable protein it's actually a very small protein that has these disulfide bonds is insulin. So insulin is shown right here. So this is just the primary structure. So insulin is actually made up of two different polypeptide chains, but they're connected covalently to each other in two spots by these disulfide bonds right here. And so that is actually really important for the overall fold of insulin and for its function within your cell. So disulfide bonds, these are sort of interesting little extra features that really are important for protein folding and are very, very common in proteins. I think it's about 30% or 25 to 30% of all proteins have at least one disulfide bond. Now, I've said it before, and I'll say it again, of course, but the overall fold of a protein is what dictates its function. So that's a whole field of biochemistry, and it's a really important part of biochemistry in general. It's the type of research I did for many, many years, was to determine the three-dimensional structure of a protein and figure out how it's doing its function in the cell. So one interesting part about this is that different parts of proteins can actually fold independently. So the same chain can actually fold independently and then give that protein more than one function or help influence its function in some way. And so proteins will often have like these functional motifs. So one half the protein will fold independently and do one job, and the other half of the protein will fold independently and do a different job. And so these are called domains. And I don't want you to get hung up on this. I know it can be a little bit confusing, but I have an example right here. So all three of these protein domains, so each of these is a single protein. So this is protein 1, protein 2, and protein 3. Now they all have this region here in aquamarine. And so this is called a phosphatase domain. You don't need to remember that. It's just that's the domain of an enzyme, of each of these enzymes. So these are all the same kind of enzymes. And what they do is they take phosphate groups off of other biomolecules. And that's important for a whole bunch of different reasons in the cell that we don't really have time to get into. So all three of these proteins have this same similar phosphatase domain that does a very similar job. And they have similar folds. You can see they got this middle region here surrounded by alpha helices with a little bit of beta sheets thrown in for good measure. But they all have similar folds, and that's because they all do very similar jobs. However, these are three very different proteins in the cell, and that has to do with these other domains that are out here. So this domain right here on this protein is a protein interaction domain. So this domain, shown right here in orange, will actually bind to other proteins and allow this part of the molecule to take a phosphate group off that protein. Now in protein number 2, this domain is a carbohydrate binding domain. And so this protein here will bind to carbohydrates, starch, and glycogen, and then remove the phosphate group from them from the phosphatase domain. And then this molecule right here, actually I think I mixed them up. So this one is the carbohydrate binding domain. This one is a lipid binding domain. So it'll actually bind to phospholipids in the membrane and then dephosphorylate them. So we have a protein phosphatase, a lipid phosphatase, and a carbohydrate phosphatase. And so they have the same phosphatase domain, but they have these different other domains that allow them to do their specific function in the cell. So don't get too hung up on that, but that is a really common feature in domains. So most proteins, I'd say, are multi-domain proteins. So they have different parts that do different functions, all of which are related to the overall function of that protein. And I mentioned it before, but there's also oftentimes what's called quaternary structure in proteins. And so that means that a fully functional protein has to have multiple folded polypeptide chains that actually come together to make the full protein. So this protein shown right here in A and B is called hemoglobin, which we've already seen. So it carries oxygen through the blood. And for it to function properly, it needs 2 of these alpha domains, which are shown in yellow, and 2 of these beta domains, which are shown in red. So those will all interact with each other to form the final functional protein. So there's another protein down here, Cre. I forget what it does, but it requires two of these two folded polypeptide chains, and I think they're actually the same for it to function. And then over here we have the SARS-CoV-2 spike protein. And so this actually, there's three copies of the spike protein that actually get expressed and fold and then interact with each other to form what's on the outside of this cell. So this is the protein that's on the outside of the actual virus. So this would be the virus, and it's going to interact with your cells and allow it to go in. So, but this is actually three copies of the same protein. So one is shown here in blue, and so it'll interact with the one shown in orange and this one shown in turquoise. And so all three, you need three copies of the same protein to make the full structure and for the virus to survive. So it's often the case that proteins have multiple what are called subunits. So these fully functional and these fully folded proteins to come together to make the protein that's fully functional. So what happens if your proteins aren't folding correctly? Is that a problem? Normally, it's not. So that'll happen pretty frequently. Let's say you have a mutation in your protein that disrupts its folding. It doesn't fold correctly. Your cells have a really complicated ways of sort of identifying those proteins and then getting rid of them. Usually they'll take them into a vesicle and sort of all the peptides, the amino acids will be degraded and then recycled to make new proteins. A lot of times, actually not just a lot of times, almost all proteins in your body also have sort of like a shelf life. Like they don't stay functional forever. They can basically be marked by the cell and then degraded, and their amino acids can be reused. So I think there's some proteins in the lens of your eye that are there from birth, but other than that, especially in things like in your intestine, there's huge amounts of cell turnover and protein turnover. So normally misfolded proteins are not a problem in the body. However, there are pathologies associated with misfolded proteins, and these can be quite common. So there's instances where misfolded proteins can actually form insoluble aggregates in your cells that can cause disease. Now, there's a lot of details not known about Alzheimer's disease, but it is known that it is related to protein misfolding. So there's this protein in your body, in everyone's body, called amyloid precursor protein. It has a certain function. Well, amyloid precursor protein can be sort of degraded to give this amyloid beta protein, which is shown here. Now, for some reason, amyloid beta protein likes to form these insoluble granules in neurons after a certain amount of time, and if you're susceptible to that sort of thing. And so these are called amyloid plaques. So each of these little spots in these neuron cells are insoluble aggregations of this misfolded amyloid beta protein. Now, what that eventually does is it kills your brain cells to the point where you have a normal brain here and you have a patient with Alzheimer's disease brain there. That's because they're having cell death associated with these misfolded proteins. So misfolded proteins is a very active branch of research and has a lot of medical applications. And a similar instance of a disease occurring from protein misfolding comes with Parkinson's. So Parkinson's you've probably heard of. It's a lot of times associated with people sort of having these tremors. So Michael J. Fox, Muhammad Ali are very famous people who suffered and Muhammad Ali actually succumbed to it in the last couple of years from Parkinson's disease. So this is related also, just like Alzheimer's, to the misfolding of several proteins, including this protein alpha-synuclein. So alpha-synuclein, which is just a normal protein in your body, but it can become misfolded and interact with other proteins that become misfolded, and then they can form these Lewy bodies in a particular part of your brain that makes this neurotransmitter dopamine. So that once those Lewy bodies start accumulating, it can kill all those cells, and then basically you have a decrease in dopamine in your brain and in your nervous system, and that's what's causing these issues. So an interesting facet of this is that they actually figured out that if you treat patients with this molecule right here, L-DOPA, which is the precursor for dopamine, that can actually alleviate a lot of the symptoms because you're sort of providing them with these patients with an external source of dopamine, so that can get into their blood, into their nervous system, and basically restore some function. And so L-DOPA is actually, I mentioned it in the previous slide, but that is actually derived from tyrosine, and tyrosine is, of course, derived from phenylalanine. So you can see how the amino acids do a lot more than just form proteins. And so these Lewy bodies and the use of L-DOPA to treat people with these neurologic pathologies was actually, there's a really good, sad movie called Awakenings from 1990 that starred Robin Williams as the neurologist Oliver Sacks, who's a very famous neurologist who also passed away a few years ago. But unfortunately, Robin Williams played Oliver Sacks in that movie. But Robin Williams, unfortunately, they found out after he died, he was suffering from Lewy body dementia. So he was actually developing dementia based on the buildup of Lewy bodies in his cells. And so the Lewy bodies are very bad. And so basically diseases of protein misfolding are very important. There's several other important ones, but those are the two main ones that you or a loved one might have encountered. All right, so now that we've talked about what proteins are, so proteins are polymers of amino acids, and how proteins fold, through secondary structure, tertiary structure, and quaternary structure into a specific three-dimensional structure, and that three-dimensional structure dictates their function. Now we can start talking about some of the functions that proteins do in your body. So proteins do pretty much every job that your body needs done. Proteins are crazy important. And a really important class of proteins are enzymes. So you've probably heard that word before, enzymes. Well, we're going to talk about what that means. So enzymes are incredibly important proteins. And what they do is they speed up chemical reactions that would otherwise occur extremely slowly. So they do this without modifying the enzyme. So an enzyme can catalyze this chemical reaction. They can speed it up over and over and over and over again without changing the enzyme. So this is absolutely essential for all the cellular functions in your body. And enzymes are really critical to life's processes. So there are billions and billions of chemical reactions going on in your body at any given time. All sorts of chemicals are being taken in and converted to other chemicals, which are converted to other chemicals, which are converted to other chemicals, which are recycled back to the main chemicals. Every single one of these chemical reactions is catalyzed by enzymes, because enzymes allow those chemical reactions to occur fast enough that they can sustain life. So there's a table here of some different enzymes. Don't have to worry about it. It doesn't even say what they did. But an interesting part is that it shows you the non-enzymatic half-life of the reaction that they catalyze. So there's this specific enzyme. It's got the really boring name of OMP decarboxylase. So this has something to do with taking a CO2 group off of a molecule called OMP, is what I'm guessing. So the half-life of this reaction that OMP catalyzes would be 78 million years. So that basically means that if you took those two chemicals, put them in a solution, it would take that chemical reaction 78 million years for that reaction to occur. When you add this enzyme, OMP decarboxylase, now that reaction will occur 39 times in a second. So it does that reaction. It takes it from 78 million years to like 0.1 seconds, basically, that it'll occur. So that's what enzymes basically do. So there's this whole table here. This enzyme takes a reaction that would take 130,000 years and does it 95 times a second. This enzyme takes a reaction that would take 69,000 years and does it basically in 0.01 seconds. and so on and so forth. And one of the fastest enzymes known, or it might be the fastest enzyme known, is this enzyme right here, carbonic anhydrase. I mentioned it in a previous lecture because it actually uses a zinc metal ion for catalysis. So using this zinc and the overall fold of this protein, this enzyme, carbonic anhydrase, is able to take CO2 and water and turn it into, I think it's bicarbonic acid. And so this is really important for regulating the pH of your blood. So this is a really important reaction. So this reaction right here, if you just mixed CO2 and water, it would eventually turn into carbonic acid in about 5 seconds for each one. If you add carbonic anhydrase, this reaction will occur 1,000,000 times per second. So this is, it's a very, very fast enzyme that can do this very quickly. And that's really important because managing the pH level of your blood is super important. So enzymes, these are proteins that are really important because they speed up chemical reactions. There's billions of chemical reactions occurring at every moment in your body, and enzymes are the proteins that catalyze those specific reactions. So how does the overall structure of an enzyme dictate what chemical reaction it's going to catalyze? So basically, how do enzymes work? So how enzymes work is that they interact with a specific chemical or a couple of chemicals called a substrate. So you have this enzyme and a substrate, and they're going to interact together to form an enzyme substrate complex. So that molecule is going to be bound to that enzyme at a specific spot. And then the enzyme is going to catalyze that catalyze that reaction, resulting in the final product, which is going to be different from the original substrate that was there. And the enzyme is going to be unchanged, so it can interact with another substrate off of that. So how do these enzymes interact with the substrate? So they interact with the substrate at a region that's in each of these enzymes called the active site. And so this is a region of the protein that has the exact correct shape to interact with its specific substrate. So here's another so this is a depiction from an actual protein. You can see it's interacting with this carbohydrate group here that's kind of long, and you see that it has this nice channel shaped right here that just is due to the structure of the protein. So if they had a different structure, it wouldn't be able to interact specifically with that particular chemical. So there's another example right here. We have a substrate and we have an enzyme, and the enzyme will sort of actually change shape a little bit, and the substrate will actually change shape a little bit, but they're completely sort of, it's like a lock and key basically between the enzyme and the substrate to form that enzyme substrate complex. And then this will be changed and released as a product. And so the active site of an enzyme is sort of the business end of the enzyme. It's this part that's actually going to react, that's going to, number one, interact with that substrate, and it's also going to catalyze that chemical reaction. So there's going to be different chemical groups at this active site that are actually going to be able to change that chemical into a product. So it's very important to understand that enzymes bind substrates at what's called an active site. Now, another really important part about enzymes that keeps biochemists and structural biologists very busy is that enzymes are highly substrate specific. So each different enzyme will interact specifically with a particular structure based on the shape of their active sites. So here we have two proteins. They each have active sites right there. So the shape and the chemical characteristics of that active site are going to dictate what exactly type of substrate that they can interact with. So chymotrypsin and trypsin are really similar enzymes, so they're called proteases. And what they do is they basically they both break these peptide bonds right here. So this is an amino acid chain. This is an amino acid chain. You have that carboxyl group, and then there's a peptide bond between the amino group of a different protein. Same with right here, carboxyl group, amino group, peptide bond, peptide bond. So proteases will every single protease does this same thing where it breaks these peptide bonds and then degrades the protein. That's really important. If you eat a steak or something, you have to have an enzyme in your body that can break the peptide bonds in the protein in that steak so you can get the amino acids and use them to make food and energy for your body. Now, the interesting difference between chymotrypsin and trypsin, though, is that they both have very specific substrate specificities. So chymotrypsin, while it breaks this amino bond right here, it'll only break the amino bond after a lysine or an arginine residue. So these are positively charged amino acids. Actually, there's a mistake there, so that I repeated the same one once. So trypsin will only break peptide bonds after a lysine or an arginine, which are positively charged. Chymotrypsin needs a phenylalanine. So that is an aromatic amino acid. Remember, it has that benzyl ring right there. So chymotrypsin will only break a peptide bond after an aromatic amino acid like phenylalanine. And this has to do with the shape of their active sites. So chymotrypsin has this specific chemical characteristics and shape that allows it to interact with a phenylalanine residue at that position and then break that peptide bond. Trypsin is different. You see it has a different amino acid here that's negatively charged, and that's going to interact with the positive charge on a lysine or an arginine. So this is a lysine right here, and then it'll break that peptide bond. So this is, sorry, I made a mistake on this slide, but this is just demonstrating that enzymes are very highly substrate specific. So the structure and the chemical characteristics of their active sites allows them to only interact with a very specific type of substrate and ignore other substrates. So if all your enzymes in your body were just willy-nilly able to interact with all sorts of substrates, it would really put your whole biological system out of whack. So this is really important feature of enzymes. So I mentioned before that, of course, there's billions and billions of chemical reactions going on in all of your cells at every single moment that you are alive. If that stops, then you cease to exist for the most part. So what are catalyzing these biological chemical reactions are enzymes. So an example of this would be metabolism of a glucose molecule. So your body has a way of ingesting glucose, and then that glucose is basically burned or oxidized into CO2, which releases, which is captured in the form of ATP energy. So that's basically the basis of all metabolism and how where you eat food and you turn that into energy. So the breakdown of this glucose doesn't happen all at once. It happens through a series of chemical transformations of this glucose molecule. So this glucose right here, a phosphate group is put on it. Its shape is changed. Another phosphate group is put on it. It's split into two molecules. Another phosphate group is put on it, and so on and so forth. So this molecule gets completely transformed from glucose into this molecule pyruvate. So that pyruvate is then converted into a molecule called acetyl-CoA, and then that acetyl-CoA enters into the citric acid cycle. And then it's continues to be changed into different molecules to release energy in the form of electrons that can be harnessed to make ATP. Now, I've just circled something like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. So I just circled 19 different chemicals that are involved in the transformation of glucose to CO2, which is released in these chemical reactions. Now, every single one of these chemical reactions, every one of these transformations of glucose from glucose to CO2 is catalyzed by a specific enzyme with a specific name and a specific way that it functions. So we have hexokinase, we have phosphoglucose isomerase, we have phosphofructokinase, we have aldolase, we have glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, enolase, pyruvate kinase, and so on and so forth. The same with the citric acid cycle. These are different enzymes catalyzing each of these specific reactions. So I hope at this point that you have sort of an understanding of what enzymes are and how they catalyze these chemical reactions. So they take one chemical, a substrate, they bind to it very specifically. They may bind to other molecules as well, and then they catalyze a chemical reaction, changing that chemical into something else, into a product. So these enzymes are very important for all of life's processes. And another way to study and to sort of use enzymes for our purposes is that enzyme inhibitors are often used as drugs. So if you can design a drug like this drug right here, which is called dorzolamide, that sort of fits into the active site, you can block the natural substrate from going into that active site, and you can inhibit that enzyme. Now, that often has a lot of really good health applications. So, for instance, this molecule, which is called dorzolamide, it's an inhibitor of that carbonic anhydrase enzyme that we discussed a little bit earlier, one of the fastest enzyme known, and that's used to treat glaucoma. So you actually have glaucoma is high pressure in the eyes, and so it can be caused by sort of this buildup of fluid. So this dorzolamide is one of the first things you give to patients who have glaucoma to sort of stop them producing so much liquid in the eyes and help alleviate some of those really painful, uncomfortable symptoms of glaucoma. So enzyme inhibitors, they're really useful for studying enzymes as well, because you can sort of see what chemicals fit into that active site really well. But they're also really useful as drug targets to modify your biological processes to treat some sort of disease or disorder. So there are actually also a lot of different proteins that are not enzymes. A lot of proteins are not enzymes, so not all proteins are enzymes. Some proteins just play a very important structural role for our cells and for our bodies. So the most abundant protein in your body is collagen. So you've probably heard of collagen before. So it provides structure and elasticity to connective tissue. So your bones, your tendons, your ligaments, and your skin. So collagen is really important. So it's so structurally stable because it's actually 3 polypeptide chains that form these really strong triple helices, as you can see here. So one of the chains is in pink, one's in blue, and one's in yellow. And so they'll interact to form these really strong fibrils, and you can build up from that. And other ones can interact. It can sort of interact with other connective tissue at the top and the bottom and stuff like that. Keratin is another similar one. So keratin is what gives your hair and your fingernails. So it's the protein component of hair and fingernails. And it also forms these sort of helical things, these amino acids that can sort of interact with each other and make these really long, strong, stable fibers. And so that's why your hair and your fingernails are so strong. They're made up of these protein fibrils. There's also structural proteins that are really important within the cell itself. So actin and tubulin are 2 of those. So actin is a protein, it's shown right here, and it can interact with other actin molecules to form these structures in the cell that actually give the cell sort of structure to it. So they stop the cell from just sort of collapsing in on itself. And so the actin is stained here in these cells in green, and you can see they form these long fibers that provide a lot of stability and movement of the cells. So tubulins are other ones. So here's an individual tubulin molecule, and it'll actually form these interactions with other tubulin molecules to make these fibers. So these fibers are really common motif in structural proteins. But proteins, apart from being enzymes, also just provide structural support to your body. So proteins are also really important for signal transduction or cell signaling. And so we'll talk a bit more about this in one of the lectures. I think one of the lectures is basically completely devoted to signal transduction, but it's a really cool topic. A lot of people get sort of scared off by it, and from looking at these figures, you can kind of see why. It gets really complicated, but the overall point of signal transduction is that if you just look at this figure right here, basically, if there's an external signal in the cell that basically where the cell needs to change its behavior or its activity or something, your body can produce molecules that will be received at the cell and then change the behavior and the activity of the cell on the inside. So this is a really huge field in biochemistry and cell biology. And so right here, see, we have, let's say this is like an insulin receptor or something. So insulin will bind to the insulin receptor. So that'll get received at the cell, and that'll cause changes actually within the cell that'll change the physiology of that cell. In the case of insulin, it'll tell the cell, hey, there's excess glucose in the bloodstream. Start making glucose transporters so you can bring glucose into the cell. So, that's actually what is shown right here. So insulin is binding to this receptor, and a bunch of different proteins in the inside all interact with each other to make a change. And then you get these glucose transporters that bring glucose into the cell. So cells also have to be able to talk to each other. So that is an example of that is right here. So cells can actually interact with each other by these cell signaling molecules, and that can be really important for immunity and just cell, anti-cancer properties of cells. This right here is another form of one of these receptors, and it sort of makes all these proteins interact, and then that causes different behaviors in the cells. So cell survival, apoptosis, that means that the cell is programmed to die. You get a signal saying this cell, you need to die. Proliferation, that can, cause telling the cell to divide basically, or differentiation. So like when you're an embryo or something, there's different signals that'll say, hey, this cell become a heart cell, this cell become a muscle cell, this cell become a skin cell. So cell signaling is, like I said, is a very big topic. But these receptors are all proteins that are embedded in the membrane, and you can see there's a lot of different complicated families of receptors that are similar but have some differences, like maybe they bind different molecules. And here's like a protein depiction of a receptor. So it would be a protein that's embedded in the membrane at this region, and then would have some region sticking into the inside of the cell and another region sticking outside of the cell so it can receive the message. So so proteins are really important for signal transduction, where there's an external stimuli that's received by the cell, and then it causes a change inside the cell. That requires the coordination of many, many different types of proteins and other molecules. And lastly, I also just wanted to mention proteins are essential to all immune activity. So foreign invade, and so this is basically, proteins are what make antigens and antibodies, which is basically how you fight off infection. So when foreign invaders come into your cell or your body, so usually bacteria, viruses, fungi, or protozoa, they will carry unique proteins on the outside of their cells, or that are released by the bacteria or virus or fungi. All of these proteins carried by the foreign invaders are collectively called antigens. So any protein that could elicit an immune response is called an antigen. So these antigens, which here's a picture of SARS-CoV-2. So it has the spike protein here on the outside of the cell. So that would be an antigen. That's actually the antigen that's used in the SARS-CoV-2 vaccine. Here's an adenovirus. So it has these groups sticking out of it. And so those will also be recognized as antigens by your immune system. So when your immune system recognizes these specific antigens, they'll produce antibodies, which are also proteins. So here's an antibody shown right here. And it will have an exact specific shape on this part of the protein that will coordinate specifically with the antigen that it's supposed to bind and neutralize, so that your body can get rid of that foreign invader. So here is another depiction. So this is actually part of the antibody here. And then here is a molecule, I think it's from SARS-CoV-2 actually, and that it interacts specifically with that antibody at that position. So both the proteins that elicit an immune response and the antibodies that are made in response to it are all proteins that are really important for your body's function. All right, so now that we've gone through proteins, we can start on the nucleic acid polymers, DNA and RNA. So these molecules, DNA and RNA, are polymers of nucleotides, which we talked about in the previous lecture. So what happens is the phosphate on the nucleotide actually links with, so this is a phosphate group right here, another phosphate group, another phosphate group. This actually links, it's connected to this sugar up here, but it will actually link to the sugar in another nucleotide. And then that nucleotide has a phosphate group that'll link to the sugar of another nucleotide, and so on and so forth. Same, so this is DNA, so it'll have its deoxyribonucleic acid, so it does not have an OH group here, and then the bases will stick out over here. Same thing with RNA. So there's a phosphate group. It's going to connect to this sugar down here, which has a phosphate group and connects to sugar down here, and so on and so forth. This is RNA. It has an OH group here because it's made out of ribose, and then the DNA bases will be, or the RNA bases or DNA bases will be in this position right here. Now, nucleic acid polymers are so important because they store and convey genetic information. So the DNA and RNA are going to be part of this central dogma where DNA is transcribed into RNA, which is translated into protein. So this is basically taking information from DNA and turning it into a specific protein amino acid sequence with RNA as the middleman. So this is the central dogma, and this is why the nucleic acid polymers, DNA and RNA, are so very important. So DNA, of course, forms what's called a double helix. So the structure of the DNA double helix was first determined by Francis Crick, James Watson, using data from Rosalind Franklin in the lab of Maurice Wilkins, and they published their results in Nature in 1953. So this is considered basically one of the best, the grandest achievements of science, and it may be the single most important discovery in all of biological sciences. So how DNA is structured is that two DNA strands pointed in opposite direction will interact with each other by the hydrogen bonds between the bases that are sticking out of that DNA. So A is going to base pair with T and form hydrogen bonds. And then C, cytosine, is going to base pair with G, guanine, and form hydrogen bonds. So we have the structure of a DNA double helix right here. And so it goes from this direction and then curves around down into this direction. The other DNA strand is going the opposite way. So it goes from this direction and then goes up in this direction. So they're oriented opposite of each other. So these phosphate groups are going to be on the outside of the DNA strand, and that's very important. They carry negative charges. They can interact with water and stuff like that. So they're on the outside of the strand. And then on the inside of the strand are those bases that are sticking out. So there's going to be one base on one strand, and it's going to interact by hydrogen bonds with the base on the other strand. And so that's going to keep going down the entire base, and it's going to turn, forming this DNA double helix. So how do those bases interact? Well, we have guanine right here, which can be shortened to just G, and cytosine, which can be shortened to C. So here is the base of guanine, and here is the base of cytosine. So the strand will be over here for this one, over here from this one. So these different groups on these bases will interact by hydrogen bonds between these different atoms. So oxygen's carrying a partial negative charge. This hydrogen's going to carry a partial positive charge, so they'll interact. Partial positive, partial negative, they'll interact. Partial positive, partial negative, these are going to interact. So G's always interact specifically with C's in the DNA double helix. The same with A and T. So A, adenine, and T, thymine, which is only in DNA, not RNA. So they'll be attached to the strand right here. And then adenine will form a hydrogen bond here. So this partial positive charge will interact with that partial negative charge, and this partial negative charge will interact with this partial positive charge to form these hydrogen bonding interactions. So these are very weak interactions, but if you have a DNA strand that's many thousands or millions of bases long, that can be pretty strong interaction, and that's what actually forms this DNA double helix. So the two strands of the helix are brought together by these hydrogen bonds between the base pairs. And G always base pairs with C, and A always base pairs with T. So if there's a G on this side of the protein, on the corresponding side, there will be a C. If there's an A on one side of the DNA double helix, in the other strand, they'll be binding to a T. So that's how the DNA double helix works. And so this is just another figure. I want to make sure that this is clear to everybody. So this strand is going in this direction from 5' to 3' end. And so this phosphate will bond with another sugar on this group, and this base is going to stick out here. This phosphate's going to bind to this sugar on the other nucleotide, and its base is going to stick out here. This phosphate is going to bind to the other sugar to make this polymer, and the base is going to stick out here. So this base is on one strand of the DNA helix, which is going this direction. The other strand is going in this direction. These bases are going to specifically hydrogen bond with each other to hold that double helix together. And so if this base right here was an A, then this base is definitely going to be a T because of what we were talking about earlier in the previous slide. If this is a G, then this base is invariably going to be a C unless there's a problem going on. If this base is a T, then this one is going to be an A. And if this one is a C, then this base is going to be a G. So that is how the DNA double helix, these two strands going in opposite directions, are able to interact with each other to form that DNA double helix. Now, these DNA polymers can get extremely large. So for the most part, except for sperm cells and egg cells, every single cell in your body, and red blood cells, sorry for the caveats, every single cell in your body houses about 3 billion bases of DNA. It's an incredibly large amount of DNA. So if you were to take one cell, one tiny little cell in your body, and stretch that DNA out, it's going to be like 2 meters long. It'll be like 6 feet. It'll be taller than most people, probably. So how are these cells able to fit all of this DNA into not even just the cell, into the nucleus? So to do this, for the cell to achieve this, what happens is this DNA double helix will actually make these really stable interactions with a bunch of different proteins. So these ones specifically are called histones, and basically the helix will wrap around all these proteins. And so here's a good description of it here. So you have the DNA double helix. So it'll wrap around these groups of histines called nucleosomes. And then these nucleosomes will form what are called like these beads on a string. And you can sort of see that in this region right here. Those beads, the nucleosomes, will then form these higher order, more compact structures. Those will get more compact, and then those will even fold onto each other. And you get a really compact form of DNA called a chromosome. So this is the general principle of how your cells are able to fit all of that genetic information into each one of your cells. Now, one of the reasons why the discovery of the structure of the DNA double helix was such a big deal was that it immediately answered a very old question in science. So how are genetic traits passed on to offspring? So Charles Darwin, way back in the 1840s, 1850s, he knew that genetic traits were passed on to offspring, and he used that as a basis for the theory of evolution. However, how those traits, how genetic information is passed on to offspring was completely unknown until the discovery of the structure of the DNA double helix. So in the paper that Watson and Crick published on the structure of the DNA double helix, They wrote this sentence at the very end of the paper. It says, "It has not escaped our notice that the specific pairing we have postulated," he means the ACGT stuff, immediately suggests a possible copying mechanism for the genetic material. And that was written in Nature, April 25th, 1953. Now, this is, in the history of science, this is considered a very coy statement. So this is really understating what they found. So the structure of the DNA double helix immediately shows you how you can replicate that strand. So we have a double-stranded DNA helix right here. Now, if you were to separate one of those strands from the other, what you could do, so if you have that strand here, just by which nucleotides are on this strand, you can immediately just copy a second strand that base pairs exactly with those other strands. And now you have another double-stranded helix. The same thing would happen over here. So you have one strand and then you copy another strand directly onto it. Those can then break up into two strands. Those can break up into two strands. And now you have two strands, and then they can be copied with another strand just by matching that base pairing of those A's, C's, G's, and T's. So I hope that sort of makes sense. So the structure of DNA, so they could have just spiked the football and be like, we know how DNA is replicated and how it's passed on to other generations. But apparently James Watson got a little nervous that maybe they'd gotten it wrong, so they put this statement in as well. Now, this copying mechanism is very simple. And so basically anybody, if they give you a sequence of a DNA strand, you can figure out how to copy it based on these this base pairing between A's, T's, C's, and G's. So if we have this sequence here, we can immediately make another copy of it. So DNA always is synthesized in 5' to 3' direction. So we have a double-stranded helix right there. You can see A's goes with T's, C's with G's throughout the entire strand. Now, if we take this bottom strand, so A, G, G, C, T, A, T, G, T, A, G, T, 5', then we can build another strand going in the opposite direction right above it. So A would go with T, G with C, C, G, A, T, T, A, C, A, T, C, A, 3'. You notice this is the exact same sequence that we got from here before. So all the information necessary to replicate one strand of DNA is encoded in the other strand of DNA. So this is a really important feature of DNA. You don't need to be able to copy a DNA sequence, but it's kind of fun and easy to practice. So if you want to do that, have at it. So that's DNA replication. It's a very important step in the field of biology. Now, I'm just going to point out quickly that DNA is replicated in our cells by a bunch of different proteins that work together to do this huge feat. In each of your cells, there's 3 billion bases of DNA. And interestingly, that can actually be replicated in about an hour. So that is a huge feat with very little mutations or no mutations, basically. So what happens is that the DNA strands are separated by a specific protein called a helicase, and then a protein called a polymerase, which is really well named because it makes the polymer of the DNA. So you have a DNA template strand. There's actually an RNA primer that has to bind first, and then this polymerase can synthesize additional DNA on that other strand. So just like what we were talking about before, the polymerase is actually what's inputting those individual nucleotides into the right position and making that second strand from the first template strand. So here is a structure of a protein structure of a polymerase here, and it actually has like this really cool structure where it's sort of got a palm region, and a thumb region, and finger regions. And then the DNA is going to be in this region right here. So it's sort of holding onto it. And then it's got this region right here. And you can see all the alpha helices and beta sheets in this structure. And so the polymerase will actually work with other proteins to separate out those strands and then synthesize new strands onto the old strand in either direction. Now, another really big breakthrough in biology was the invention of a technique called the polymerase chain reaction, or PCR, which you may have heard of before. So this was actually invented by this guy, Kary Mullis, in 1983. And what this technique does is it allows you to replicate a specific chunk of DNA from, if you have some DNA, you can replicate it a bunch of times simply by putting that DNA in a tube, combining it with enzymes, nucleotides, and primers, and then basically you just put it on a heat block that heats the DNA and then cools it. So this can make a bunch of different copies of DNA, and this has really ushered in a complete revolution in biology. So there's a million different applications for this technology, which is quite simple. So to walk you through how it works, this is what you would start with. So you have your DNA region that you want to replicate. So let's say you found a woolly mammoth in the Siberian ice and you want to try and copy some of its DNA to see what it looks like. Well, you just take any DNA that might be found in that woolly mammoth, you mix it with a polymerase, which would actually, copy the DNA. You mix it with nucleotides, because that's what's going to, they're called DNTPs here, so deoxynucleic triphosphates. It just means nucleotides. So this is what's actually going to build it up. And then you have some primers. So I'll get to those in a second. So what you do is you heat the sample, and so that'll separate the DNA strands, because remember, they're held together by these hydrogen bonds between the A, C's, G's, and T's, so you can just break those by heating. So then you take your primers, and your primers are going to be specific for the region of DNA that you want to amplify, and so they'll go on one side and the other side. Oops, sorry about that. And then the polymerase will come and it'll find that primer and it'll copy that DNA. It'll put these building blocks into it and the primer will copy that DNA. And so now you had one strand of that DNA, now you have two strands of the DNA, and same with down here. So you can keep doing that. So you just repeat those cycles. So you just heat the DNA up to separate the two strands. Then you allow those primers to anneal to it, and then you lower the temperature and allow the polymerase to extend it further. And so by the second, if you just start with one piece of DNA, by the first cycle, you'd have 2 pieces of DNA, then you'd have 4 pieces of DNA, then you'd have 8 pieces of DNA, then you'd have 16 pieces of DNA, then you'd have 32 pieces of DNA, and then 64, and so on and so forth, to the point where you have, if you do like 30 cycles of this, which is the normal amount of cycles you would do in the lab, you have a ton of DNA. So this is really important for identifying different genes. It's important for cloning, for making proteins. It's important for learning things about the genetics of animals like woolly mammoths, or, you can find out what kind of diseases King Tut had from sequencing the DNA, determining the DNA from the mummy. So PCR has a million different applications, and it's a really important technique in the lab. And so if we're talking about PCR, we might as well mention a little bit about COVID-19 tests. So if you're sick and you think that you might have COVID, it's probably a good idea to get tested so you don't spread it to other people. And there's basically 2 choices for which kind of COVID-19 test you would have. So one is PCR-based, like we just talked about, and one is antigen-based, like we talked about earlier in the lecture. So the same thing is going to happen for the collection of each of these tests. So someone's going to stick a swab in your nose and collect some material from your nasal cavity. So with the PCR test, what the PCR test does is it will detect viral RNA in that sample. And so it'll do that because you design primers that will amplify through PCR that specific DNA sequence that is in the SARS-CoV-2 genome, but not in your genome. So it can distinguish between the viral genome and your genome. Now, this test actually has a little bit of a modification to it. So when this polymerase is actually making a new thing of DNA, that primer, there'll actually be a primer in the middle as well that has a fluorescent signal. And so when it's making that DNA, it'll break off that signal and this will give a little light flash. Now, when you get enough DNA, let's say you start getting like thousands and thousands of DNA after like the 15th to 20th cycle, if you start getting a slight signal, that means that there is SARS-CoV-2 in that sample. So that is the PCR, also called the qPCR or RT-qPCR test, to test for COVID in a biological sample. So that test is very sensitive and very specific. So it was only going to copy viral DNA. And some of these types of tests are so specific that even if there's one viral genome in that sample, it will detect it. And so that's why you could not be shedding virus out of your nose, but still test positive for COVID for a long time. However, the drawback from this test is that it takes some time. You have to set it up. It takes like 40 minutes to an hour to go through all 30 cycles. So that takes some time. You're not going to get your results back for a little bit. There are some hybrid tests that are a little quicker, but that's not super important. So the antigen test works differently. So what the antigen test does is it's testing for antigens. So this would be the spike protein on the outside of SARS-CoV-2. So if you have virus in your nose, they're going to have that spike protein in there. So what this test does, it actually works. It's a modification of an ELISA, or it is just an ELISA test, I think. And so it works just like a pregnancy test. So if there is SARS-CoV-2 there are going to be antibodies that are going to bind that specific antigen. And if they do, then that region right there is going to change color and give you a positive test. So this will actually detect viral proteins rather than the viral RNA or the viral genome. So that the antigen test is way quicker because you can just take it, throw it in the thing. It takes 5-10 minutes. And then you know if the antigen's bound to the antibody or not. But it is a little bit less specific because there's going to be a lot less of the antigen in your nasal cavity than the viral genome. So those are the two main COVID-19 tests, PCR-based and antigen-based. And one thing I want to mention very quickly is that we talked about how important or how big of a feat it is really that your body is able to replicate these 3 billion bases of DNA in just an hour without making very many mistakes. So the reason why there's not a lot of mistakes made is that the integrity of your viral genome, your genome is very important. So any mutations that occur or mistakes that occur during replication can be passed on, can lower your fitness and your survival and be passed on to your offspring. So human beings and all other organisms have evolved these proteins that function basically to have some sort of error correction in your genome. So the polymerase itself has an error correction mechanism. So if the wrong base gets put in, the DNA kind of flops around and then it'll hit this exonuclease region, which is shown right here in this cartoon. See, this is like the palm. This is the fingers. This is the thumb. So if the wrong base gets put in, the DNA strand will kind of become wobbly, and then it'll reach this exonuclease site, and it'll kick off that one and put the correct one in its place. There's also a bunch of proteins that basically scan the genome looking for errors, and then they can cut out that section of DNA, and another polymerase can come in and put the correct bases in. So this error correction is really important to maintain the integrity of your genome, although errors do definitely occur. And mutations also can just occur not during replication, just being exposed to things like UV light or different chemicals. So these sort of mutagens or carcinogens, that's sort of what they're doing. They're messing with the sequence of your DNA bases and the base pairing. So this brings us to the question of what is a gene? So you've probably heard the term gene before. I've been throwing around the term genetic and genome all over the place. So of the 3 billion DNA bases that are in the human genome, right here depicted as chromosomes, Actually, only a very small amount of those are considered genes. So, what a gene is, it's a region, a small region of DNA that is actually transcribed into the molecule RNA. So, it's a very specific region of that DNA where basically something called an RNA polymerase is going to come in, separate that strand, and then make an RNA polymer from that sequence on that DNA. So most of the DNA in your body is non-coding DNA. So it doesn't code for any RNA and it's not considered a gene. The function of all this extra DNA, that is something that's still being worked out from people. But gene sequence are really what dictates your phenotype. And what a phenotype is an inheritable trait in an organism. And this is related to the fact that these specific genes are going to be transcribed into RNA that are going to be translated into proteins. So what type of proteins, based on the information in your genes, get expressed has to do with the specific sequence on those genes. So when we talk about heritability, we talked about that a little bit in the last lecture. So both your parents can have a mutated gene in one specific region. There's a 1 in 4 chance where you'll inherit both the good versions of the gene, 50-50 chance that you'll be a carrier but not affected because you have one copy of the good gene. But then there's a 1 in 4 chance that you're going to inherit both bad forms of the gene from your parents. So a gene is the specific regions of DNA that are going to be transcribed into RNA, which is going to be translated into protein. Most of your DNA are not gene sequences. So inherited diseases, those basically originate as mutations that affect protein function in your DNA. So mutations in your specific genes can result in a change in that protein sequence that can affect its function. So for instance of this is the disease osteogenesis imperfecta, which is often called brittle bone disease. Now, humans can live for many years and live very successful lives, like this guy, Dr. Peter Radke, with this disease osteogenesis imperfecta, but it definitely does affect quality of life. So you're always breaking bones and stuff like that. And the reason why this inherited disease originates is because it's often caused by mutation in the gene sequences that encode for an important glycine amino acids in collagen. So we already looked at collagen very quickly when we were talking about structural proteins. It's made of these 3 triple helices that interact to make a really strong collagen molecule that keeps your tendons strong and your bones strong and your skin strong. So the interaction between these different helices has to do a lot with these glycine residues. You can see there's repeating glycine residues in the sequence of the collagen amino acid sequence. So in many individuals with osteogenesis imperfecta, this glycine residue is mutated in the gene, and then that eventually creates a protein that doesn't have a glycine in that position, and then it weakens the collagen in your body, and you develop something like osteogenesis imperfecta. So we'll talk more about inherited diseases later in the course, but I just wanted to mention it since we were talking about DNA and genes. So the process of transcribing an RNA or making an RNA polymer from a DNA sequence is called transcription. So just like DNA is replicated using one strand as the template, the sequence of an RNA polymer is transcribed from a gene in the DNA genome using that base pairing information. So it's also formed in the 5' to 3' direction, just like DNA, but RNA has uracil instead of thymine. And so this brings us back to our central dogma, where DNA is transcribed into an RNA message, which is then translated into a specific protein molecule. So we can actually really easily, so if you had, for instance, this DNA sequence, you could already determine what the RNA sequence is going to be from that. So RNA is going to be built in the 5' to 3' direction. And so we have an A right here. That would normally base pair with T, but there is no T in RNA, so this will be a U. So then it'll be C, C, G, A, U, U, A, C, A, U, C, A, G, A. And so this would be the RNA sequence that would be transcribed from that specific DNA sequence. And you see that it keeps all the information in the gene. It's just in the opposite orientation, and there are U's instead of T's. So it is faithfully replicating that information in the DNA into an RNA molecule. So there are actually 3 different kinds of RNA that are made by your cell through transcription. So one of them is called messenger RNA or mRNA, and that's shown right here. So this is it's the message. So it's the RNA sequence that's actually going to get translated into a protein. There's also another kind called ribosomal RNA or rRNA. And so this RNA will actually fold and interact with proteins in a 3-dimensional structure, and it's involved in the translation of this messenger RNA. A third one is called transfer RNA. And so that is shown right here. So the transfer RNA is also involved in the translation of the messenger RNA. So it actually has 3 different parts. So it'll correspond to specific regions on this message, and it's what's actually going to carry that amino acid to the specific mRNA to make that polypeptide chain. So there's 3 types of RNA. Not super important for you to remember that, but it is important in how this message from the messenger RNA gets translated into a protein sequence. So through the interaction of that messenger RNA, the transfer RNA, and the ribosomal RNA in the form of a ribosome, the mRNA strand is going to be translated into a specific protein amino acid sequence. So this is going to be based on something called the genetic code. So in this mRNA strand, there's going to be a specific group of codes that are on this strand. So then those transfer RNAs, this mRNA is going to get fed into the ribosome, and these transfer RNAs are going to have the specific code that matches that code on the mRNA, and then carry a specific amino acid that's associated with that 3-letter code. So it does get a little confusing, but don't get too lost in the weeds into it. Basically, what it means is that each of the 3 codes on the mRNA sequence are going to correspond to a specific amino acid. And so all of this has been worked up. So right here, we have a 3-letter code. So it's going to be CCU on the mRNA. And so which amino acid is that going to carry to that specific position? So we can figure that out from this genetic code table right here. So CCU, that is going to be a proline amino acid. And so that transfer RNA will bring that amino acid there, and then the next code will bring another amino acid, and they'll form a peptide bond in this region from the polymerase, or from the ribosome actually, and they will form that peptide bond that forms the protein. So this is how you take that message from DNA, transcribe it into RNA, and this is how that RNA message is translated into a specific protein sequence. So here's another depiction of how that's working. So we have an mRNA strand that's interacting with a ribosome here, and then the transfer RNAs are going to bring those specific amino acids in there. So we can actually practice translating the following, this mRNA or this RNA transcript here, mRNA transcript. And so we can do that by breaking it up into 3-letter codes and seeing which amino acid that interacts with. So for this first one, UCC, we have UCC, and so that's going to be a serine residue. So the 1-letter code would be S. Then we have UGA, so UGA. UGU, I got confused. So UGU, so that's cysteine, so that's a C. There's also some, I should mention, there are some stop codons. So it'll tell, if those codons are hit, then it'll tell it to stop making a protein. That's just an aside. We have AUC next, so that's an isoleucine. We have a GAG, so that's a glutamate, which is actually an E for the 1-letter code. We have an AAU, and so that's an asparagine, so the one letter code is actually an N. We have a UGC, which is another cysteine, and then we have GAA. And so that gives us another glutamate. And so the amino acid sequence of ours spells out science. So that is how that information in the mRNA sequence is actually translated into a specific protein sequence or primary protein sequence. So the way that prokaryotes like bacteria and eukaryotes like mammalian cells, the way that they express their genetic information from DNA to RNA to protein works in pretty much the exact same way. There's some little bit of differences. For instance, prokaryotic genomes are circular, whereas eukaryotic genomes are linear. They don't connect at the ends. But a big difference is that in prokaryotes that don't have a nuclear membrane, they can actually transcribe an mRNA sequence and make the proteins at the same time using these ribosomes here. So in eukaryotes, you can't do that. You basically have to make these mRNA transcripts. They're actually processed in eukaryotes. Then they have to be exported out of the nucleus, and that's where the ribosomes are that are going to make the protein sequences from that mRNA transcript. So protein expression in prokaryotes and eukaryotes is very similar, but there are some important differences. So now moving on to carbohydrates. So carbohydrates can also form really long polymers called polysaccharides. So polysaccharides are polymers of simple sugars. The most important ones are just polymers of glucose. So glucose, which is a simple sugar or monosaccharide, can form extremely long polymers called polysaccharides. So the most important ones are cellulose, starch, glycogen, and chitin, which isn't listed here on this table. So these are the most abundant of the polysaccharides. And basically what they are they're made up of different arrangements of glucose polymers. So they're all made entirely of glucose polymers. Cellulose is found in plants. Starch is found in plants. Chitin is found in insects and fungi. And then glycogen is found in animal cells. So the glucose subunits, they're all made out of glucose, but they can either be alpha glucose or beta glucose. And I'll talk more about that in a minute. And then they can have different bonds of different positions where the carbohydrate is attached, where they're connected at. So the carbon atoms are actually numbered. So there's 1, 2, 3, 4, 5, 6 carbons in glucose. So for instance, in cellulose, the glucose molecules are linked by 1 to 4. So this carbon will be linked to the 4 carbon on another glucose molecule. That's very common. Sometimes there can be 1 to 6, where this 1 carbon is linked to this number 6 glucose. And that actually makes branches in these molecules. So cellulose does not have branches. Amylose, which is a part of starch, does not have branches. Amylopectin in starch does have branches, and glycogen has branches. And you can see sort of how these are arranged. That has to do with the sort of form that they take. So amylopectin will have these branches off the side. Glycogen will also have the branches off the side. And they'll make these different shapes. So cellulose will make really strong fibers. Amylose will form these helical structures. Amylopectin will form these branch structures. They actually do make helices as well around these molecules. And then glycogen forms a really soluble sort of branched molecule. So these are the different polysaccharides, which are polymers of glucose. So an important distinction between which type of polysaccharide you're making is which type of glucose molecule is involved and then how they're linked together. So glucose molecules, as I said, the carbons are numbered 1, 2, 3, 4, 5, 6. And then glucose specifically can have 2 different orientations at this position right here. So if this OH group at the 1 carbon is pointed down, that is an alpha anomer. And if this this hydroxyl group off of the one carbon is pointed up, that would be the beta anomer. And so this is actually really important because something like cellulose is made up of beta glucose linkages, and that molecule is completely unedible or indigestible by us. It's basically the reason why you can't eat grass or newspaper and get any nutrients from them. They're made up of glucose, but they're made of a form of glucose, this beta anomer here, that can't be digested by our bodies. Now, conversely, we can eat starch and I guess glycogen we can sort of break down for our own purposes because those are made from the alpha anomer. So even a small chemical change like this can really change the properties of a molecule and how you're able to metabolize it. So these alpha glucose molecules will be linked in starch and glycogen in this way right here. So you'll have a long chain of alpha 1,4 linkages. And you can see that makes sense because the 1 carbon is connected to the 4 carbon. So these will be connected and a water will actually be lost. So you can see that alpha 1,4, alpha 1,4, alpha 1,4, alpha 1,4, so on and so forth down the chain. But glycogen and amylopectin in starch, they can actually have also alpha 1,6 linkages. So this is an alpha 1,6 linkage here. And so what that does is it increases the amount of glucose that you can fit onto this polymer because then you can continue that chain through these alpha 1,4 linkages that are shown right here. So this is a really important way to pack as much glucose into a molecule like starch or glycogen as possible by adding these alpha 1,6 branches. So glycogen is the storage form of glucose in animals. So as I said, it's a large polysaccharide and it's formed from these alpha 1,4 linked glucose chains. So this will continue on with these alpha 1,6 branches at it's about every 10 to 11 units. And then this would continue on. And then there'd be another branch at 10 to 11 units, another branch every 10 to 11 units. It actually does form sort of a helical structure when you're looking at it in 3 dimensions, but that's not super important. The one thing that's really important is that glycogen is a soluble form of a glucose polymer. So it makes these soluble granules, which can be visualized like this or visualized like this with the individual glucose units. So this is stored in a soluble form because if you start exercising or something, you want to break down that glycogen really quickly. Or if your blood glucose level is getting a bit too low, you want to break down that glucose from that glycogen very quickly so you can export it into the blood. So glycogen is a really useful form of glucose storage for animals that might have to, you're a gazelle and then a lion is attacking you. You might have to get away very quickly. And so this glycogen provides energy in the form of glucose to do that. Now glycogen is a really good energy storage form, but it really only works in the short term. So your glycogen stores are going to be depleted pretty quickly. So this right here is a graph showing the amount of glycogen in a muscle cell during exercise. So someone's exercising at 90% effort for an hour. Well, you start with a lot of glycogen here, but as you're exercising, that depletes very quickly to where after an hour after you've exercised, only about 5% of that glycogen remains. So glycogen is a really important storage form of glucose in animals, and it can be mobilized very quickly because it's soluble to provide energy in short bursts. So starch is sort of, it's similar to glycogen, but it's the storage form of glucose in plants. So it's actually formed of 2 components. One is the minor component is amylose, which is just these alpha 1,4 linked chains, so that would continue. And then the second one is amylopectin, and that is just like glycogen. It has these alpha 1,4 chains with these alpha 1,6 branches. However, these branches are less frequent. So the branches in amylopectin and in starch are about every 30 units. Now, this has some biophysical properties to it. So the fact that there's less branches actually allows amylopectin to form these really stable helical structures. So this is one glucose chain, and then there was a branch, and then this is an adjacent glucose chain right here. And so they can actually form helical structures and then sort of bunch together really closely. Now, what's the point of that? Well, this actually makes starch insoluble in water. So if you've ever tried putting like cornstarch into water, it sort of just makes a glue. It doesn't dissolve very easily. And this is because plants really don't need to mobilize their glucose storages to like run away from a lion or something. Plants aren't unable to do that. There's basically two reasons plants use starch. One is stored in endosperms, like in wheat, or in tubers like potatoes, and those are for like to keep the seeds healthy and stuff like that. The other reason plants use starch is for their diurnal metabolism. So plants, at the end of the day, their leaves will be full of starch, and you can actually stain starch using iodine. So lots of starch at the end of the day. They've been doing photosynthesis. They've been building up starch in their leaves. However, during the night, there's no photosynthesis happening. So that's why they will deplete the starch from the leaves to provide energy for the plant so it can keep growing. So these crystalline forms of glucose storage allow plants to store a lot of glucose in a really small amount of space, but it's highly insoluble and made in these granules. So glucose, the storage form in plants, is starch, and they use it for energy, and it's a little bit different than glycogen, but also has some similarities. Now, another really important glucose polymer is cellulose. So cellulose is a polysaccharide in plants that doesn't provide energy for the cell. Instead, it has a structural function. So cellulose is just made of these chains of beta-1,4 glycosidic linkages. So these glucose molecules are linked in the beta form, so not the alpha form. And they just make these single long chains with no branches, but it makes a lot of these chains, and they can sort of interact with each other. We sort of saw this in proteins with like keratin and collagen. So they can make these really, really long fibrils that are held together by hydrogen bonds, and makes a really stable structure. So that's actually what gives plants like, grass and other things, and like stems and leaves, what gives them their structural integrity. So cellulose is one of the most abundant organic compounds on Earth, and we use it all the time to make like newspaper and other types of paper and stuff like that. So cellulose is really important. So because it's these beta forms of glucose that are linked together, we're not able to digest cellulose. However, other organisms like cows and fermenters in their gut, they have microorganisms in their guts that are able to break down that beta anomer of cellulose. So then we can eat the cows and get the energy from it indirectly. So that's cellulose. It's not an energy form of glucose. It provides actually structural function. Cellulose does. And so the last glucose polymer that we're going to talk about is actually a glucosamine polymer called chitin. So chitin is a polysaccharide made from beta-1,4 linkages of a modified glucose molecule called glucosamine. So glucosamine has this amine group here. So this is glucosamine right here. But even though the glucose is modified in this is a really important carbohydrate polymer. And it's about the second most abundant polysaccharide on the planet after cellulose. And that's because it forms the cell wall in fungi, and then it also forms the exoskeleton in insects and in arthropods, so insects and crustaceans. So that's chitin. It's just a different storage form of a glucose polymer. So there's also other kinds of glucose polymers that don't necessarily have glucose in them. They can, but one of those ones that's really important is called protein glycosylation. So proteins can actually be modified by these groups of polysaccharides. So these different types of carbohydrate groups, these simple sugars, like there's other ones called galactose we've mentioned, galactosamine, N-acetylglucosamine. We talked about glucosamine. There's also another one called mannose, N-acetylmanosamine, fucose, and sialic acid. These are all different carbohydrates, and they can actually decorate the outside of proteins. So certain amino acids can be modified. One of them is asparagine, which ASN right here. They can be modified with these groups of carbohydrates. So this may seem like just an aside. Oh, you throw in some carbohydrates on a protein. Well, it's actually really important. So about 50% of all proteins at least have one carbohydrate group on them. So they're called glycoproteins. And this can significantly change the behavior and properties of a protein. So this protein right here is called erythropoietin. So it's a signaling molecule in your blood. Actually tells your white blood cells, red blood cells to divide if they're too low. It is way more stable in your bloodstream if it's glycosylated. So if you take off all these glucose groups, which you can totally do that through chemical methods, the protein's only going to survive at a way less amount of time than if it is glycosylated. So carbohydrates can also be added to protein to increase their sort of functionality, and this is very common amongst different proteins. So this protein glycosylation is also involved in something that you're probably familiar with, which is blood groups. So in humans, we have A, B, and O blood groups, and then different, subgroups off of that. Though what those are based on are protein glycosylation patterns on the outside of red blood cells. So if you have the O antigen, you would have a glycosylation pattern on the red blood cell proteins outside the red blood cell that's shaped like this. If you have the A blood group, it's going to have this extra group over here. And if you have the B blood group, it's going to have an extra position over there. So this is really important because if your blood cells, let's say you have B group blood, and you give that blood to someone who has A antigen blood, the immune cells of that person who got the blood transfusion are going to recognize this carbohydrate group as a foreign invader. That's why it's referred to as an antigen, because remember, antibodies interact with antigen proteins. And so if this blood is given to this person, they may attack those red blood cells because they think it's like a bacteria or a protozoa that's in the cell. So these protein glycosylation patterns are form the basis of the human ABO blood groups. And lastly, another really important thing with protein glycosylation is it's really commonly involved in viral cell entry. So viruses usually have these proteins, or I think they kind of always have these proteins on the outside of the cell. And so those are very often glycosylated. And so how the virus is going to get into the cell is they're going to sort of hijack your machinery that's going to recognize those glycosylation patterns on that virus. And then they're going to get taken into the cell. And then they can release their genetic material and replicate. So when these virus proteins replicate, so they basically get your cell to make the viral proteins, and then they actually bud off from the membrane. And so they'll have a new thing of glycosylated proteins to invade another cell or to spread to another person and invade their cells. So this glycosylation of these viral proteins is really important to virology, and it's a feature of both influenza and SARS-CoV-2. So now that brings us to the lipids. So lipids don't really polymerize, but they do form membranes, which can kind of be seen as an aggregation of lipids or something similar to lipid polymers. So we talked about in the previous lecture about phospholipids. So phospholipids, a really important feature of them is that they're both hydrophilic and hydrophobic. So here is phosphatidylserine. We see we have the group over here. It's got some positive charges and some negative charges. So this is hydrophilic. And then, of course, these lipids all have these long hydrocarbon chains. So those are going to be hydrophobic. So it can be depicted in this way right here. So you have a hydrophilic head group and a hydrophobic tail. So that's going to be part of all lipids, but especially most lipids, but really important in phospholipids. So there's also a shorthand depiction. So this is hydrophilic. And then the tail would be hydrophobic. So these molecules can interact with both polar and nonpolar molecules, and that's really important for membrane formation. So if lipids like phospholipids are put into a polar solution like water, so just an aqueous solution, so water has, a positive charge and a negative charge. Lipids, when they're put into solutions like that, they'll spontaneously form either micelles or these things called lipid bilayers. So that's sort of shown right here and right here. So these lipids will, without any external energy, they will spontaneously form these lipid bilayers here, where the positively charged, the polar hydrophobic head groups are going to interact with the water outside the membrane and interact with the water inside the membrane. So this is a blown up version of this. There's another depiction here. So these blue molecules are all water molecules. And then you have the hydrophilic head groups up here, the hydrophilic head groups up here interacting with the water. And then the hydrophobic tails will interact with each other in that lipid bilayer membrane. So this happens spontaneously. So the main forces, there's a lot of different forces driving this. The main one is the hydrophobic effect. So that's actually an entropy-driven process. We don't need to go into that. But basically, it's just the effect that nonpolar molecules are going to interact with each other in an unaqueous environment. So that's going to drive that force. There's also other types of interactions that are involved in this. But the important part is that these lipids, because they have a polar group and a nonpolar group, will spontaneously form these bilayer membranes. So each one of your cells is going to contain many different lipid membranes. And so what those lipid membranes do is that they isolate the inside environment from the outside. So this is the main definition of a cell, basically. So it's just aqueous solution that's surrounded by a lipid membrane that contains that internal cellular environment away from that external cellular environment. So the overall cell has a lipid membrane. So this is just called like the cell membrane usually. But a bunch of different organelles within the cell also have lipid membranes. So the nucleus has a lipid bilayer. This lipid bilayer is actually continuous with the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The Golgi apparatus, which is shown right here, and we'll talk more about all of these in the next lecture, that's surrounded by a lipid bilayer. Any vacuoles that are in the cell, that's more important in plant cells, but those have a lipid membrane. So different vesicles within the cell, so peroxisomes, secretory vesicles, lysosomes, those are all going to be surrounded by a lipid bilayer. And then mitochondria as well, which we'll talk about later in more detail. Those actually have a double lipid membrane. So these lipid membranes are really important for the cell, and they keep things contained within a certain environment and then protect them against the external environment. Now, a really important feature of that is that these lipid membranes in your cells are not only made of lipids. So that's the primary structure of them, of course, but there's a lot of proteins that are embedded within these membranes. And so we sort of talked about membrane proteins a little bit. So these amino acids in these regions right here are going to be hydrophobic amino acids, so they can exist very peacefully within that lipid bilayer membrane. So proteins are really important, and the amount of proteins in different membranes varies between cells. So in the average cell that you're going to run across, let's say a liver cell or a heart cell, it's going to be probably somewhere around 50% of the membrane is going to be made up of different proteins, and the rest is just going to be a bunch of lipids. We'll talk about myelin in a minute around neurons. That can be kind of low as 25%. protein component, so it's mostly made up of lipids, and then your mitochondrial membrane, because the mitochondria are doing all of the metabolism, they need a lot of proteins to do that metabolism. So their protein component in mitochondrial membranes can be as high as like 75%. So there's a big range. And then, of course, these proteins can also be glycosylated. And then, honestly, the lipids within the membrane can be glycosylated as well. There's also, I don't really talk about it in this lecture, I kind of ran out of time, but cholesterol is also associated with these lipid membranes in animal cells, and that'll just change the fluidity of the membrane. But proteins are embedded within these membranes. So what are these proteins in the membrane doing? Well, one really important part that proteins play in the lipid membrane is they allow the cell to control the environment of the inside of the cell versus the outside of the cell. So the cell membrane, the lipids, because it's so hydrophobic, it's really hard for most compounds, especially large compounds or polar compounds, to actually just move through the cell without any external help. So molecules like that can't cross this lipid bilayer. But your cell may want to bring in a bunch of molecules that need to do some job. They may want to control the pH or the charge of the cell inside versus outside. And so proteins that are embedded in the membrane can perform these tasks. So you see here, there's some channels that allow certain molecules to go through. There's also more active carrier molecules that are sort of actually use energy to shift molecules from one side to the other. And a really important one is in your neurons. So a really important point of a lot of cell membranes, especially in your neurons, is that they generally tend to pump sodium outside of the cell and then pump potassium inside the cell. And they sort of do this in a way so the overall charge of the cell, of the inside of the cell, is different than the outside of the cell. So that's really important for all cells and especially for neurons. So these different proteins within the membrane can actually control what molecules get in and out of the cell. So this movement of ions inside and outside of the cell is really fundamental to how your neurons work. So neurons require constant transport of ions across the membrane. So in there's sort of a GIF right here. So in the resting state, the outside of the cell is going to be positively charged, and then the inside of the cell, especially the axon of the neuron, which is like the long arm part, is negatively charged. So if this cell needs to send a message to another cell, it can basically make an electrical charge that moves through that cell by bringing in positively charged ions into the cell. And you can sort of watch that right here. These positively charged ions are moving through the cell, and then that will eventually cause a change that might excite another neuron or inhibit another neuron. It gets really complicated. But this is all based, so all your neurology is based on the ability for these neurons to transport ions across the cell membrane. And so neurons actually also have another membrane that'll sort of wrap itself around the axons called a myelin sheath. So this actually increases the speed at which that electrical current can be transmitted across that axon. So it actually works as like an insulator, just like with, electrical wires in your house or whatever. So here is a neuron here. You can see there's these myelin sheaths, and these are basically just specialized cells that have a membrane that sort of wraps around the axon. And you can sort of see that here, these sort of layers of membranes that surround that axon, which is itself another membrane. So this is really important, and this is sort of outlined in this GIF right here. You can see how quickly the signal is moved through a neuron that has these myelin sheaths, and then it goes much more slowly in a neuron that does not have these myelin sheaths and oftentimes can't even reach the end of the neuron. So this causes a huge problem in people when their myelin starts to degrade. So there's a lot of different demyelinization disorders. One very common one is called multiple sclerosis that you've probably heard. So this occurs because the myelin around these neurons becomes degraded, And so now your neurons aren't able to fire properly, and it messes with your motor skills and a lot of just basic functioning of your neurology. So membranes upon membranes. So that's a really important way that neurons work. So I mentioned earlier in the lecture, I talked a bit about cell signaling and signal transduction. Well, that all occurs because of these lipid membranes, and it occurs in congruence with these lipid membranes. So the whole point of cell signaling is that there is a signal on the outside of the cell that needs to be expressed to that cell and then change the response of the cell. If you look at right here, this is sort of a very simple sort of diagram on how that works. So your cell, in response to some stimuli, let's say, it's a fight or flight response, your body is free, you see a lion and you start freaking out. Your body's going to dump epinephrine into your bloodstream. That's what these molecules would be. That epinephrine will then be received by a receptor that's embedded in that cell membrane. So it has an outside part that's going to bind that epinephrine. It's got a part that's embedded in the membrane, and then it has an inside part that's going to change once that receptor binds that cell. So that message can then be put inside the cell and result in a response to the cell. So cell signaling and the membrane of the cell are intricately linked to each other. It's also really important for neurons. We were just talking about these action potentials that get pushed through neurons. The ultimate result of that is the release of these neurotransmitters at the end of that neuron. And those neurotransmitters are then received by these same receptors as we see over here in that cell. And so that cell will then have a change inside if they have certain neurotransmitters. So cell signaling is very closely linked to lipid membranes. And so another example of this, which we'll talk about more with nutrition, is insulin signaling. So insulin signaling is a really good example of a signal transduction event in the cell. So when your blood sugar gets too high, so right after a meal, your body will release, or your pancreas will release this molecule insulin. So that will be received by a receptor at the cell, and it'll cause an intracellular change that sort of gets filtered through all changes in the activity of all these different proteins. And ultimately what happens is that this will cause proteins to translocate these glucose transporters. Remember, we talked about how important transporters are just a minute ago. And now, because there's excess glucose in the blood, this signal tells these glucose transporters to go to the cell membrane and bring in this glucose. And then that glucose can be converted into glycogen because you have extra glucose, and you should store that away for the rainy day. So you don't need to understand everything about insulin signaling, but it does sort of bring into focus what we've been talking about with proteins, the cell membrane, cell signaling, and carbohydrate polymers like glycogen. So that's insulin signaling. We'll talk a bit more about it when we talk about nutrition. All right, so that was lecture number 4, complex biomolecules. So we went through proteins, nucleic acid polymers, DNA and RNA, carbohydrates, so the complex glucose polysaccharides, and then we went through lipids and cell membranes. So I understand there was a lot of information in this lecture. And so if you don't understand something, that's not a big deal. You don't need to remember it for a test. And you can always ask me questions if you're curious about a specific topic. So I hope you enjoyed it. Please remember to finish your quiz and your short essay this week. So the short essay this week will probably make it a little simpler. Just what did you find interesting? What did you find confusing about these different topics? So I hope you enjoyed it. And as always, please let me know if you have any questions.